So I'm going to start off from the beginning. Okay. So to motivate it, I'm going to do this animation again. So we all know that we can drive when we see a green signal and stop driving when we see a red signal. That is due to that's due to the fact that we have learned that we can associate green with the action of going or hitting the accelerator, and the red, arbitrary visual symbol red, with the action of stopping or hitting the brake. As I mentioned before, humans and animals are really good at making these associations. And one type of this association is associating a visual symbol with movement, which is visuomotor associative learning. I'm interested in how that learning itself happens and not what happens after the learning happens. So I'm going to cheat a little bit and put a human brain, but all this work has been done in monkeys. Work by Earl Miller and others have demonstrated that the prefrontal cortex and basal ganglia, interconnected, are really involved in learning these visuomotor associations. But I'm interested in the cerebellum, and cerebellum has this beautiful repeating architecture that we all know about, and it has information about sensory motor representation. And it can also function as an internal model. We know that from years of literature. So that is a good candidate for learning visuomotor associations. But traditionally, cerebellum has been thought about as a motor control system, merely involved in correcting motor errors and optimizing the goal-directed movements. But cognitive-related aspects of cerebellum is not really talked about in the public. So we wanted to study if cerebellum is indeed involved in cognition. One good argument for this is the anatomy. The recent work by Peter Strick and his colleagues have shown that mid-lateral cerebellum, a specific part of the cerebellum, which they have shown that this region is connected to the prefrontal cortex, the same region which is involved in the cognitive loop. And also there is an additional loop between the basal ganglia and the mid-lateral cerebellum. So this anatomical evidence creates speculation for functional connectivity. So we used a very simple visuomotor associative learning task to study these properties, where we expect the monkeys to grab onto two bars for about 800 milliseconds. And after that, one of the two visual symbols appear. So green means he has to release its left hand, and pink means he has to release its right hand. That is the task. So just one visual symbol, and then a movement associated with that visual symbol. So as soon as he makes a correct movement, he gets the reward instantly. So we trained this monkey on this particular task for about 2 years, so he's overtrained. He always knows what green means. He always knows what pink means. So this is an overtrained condition. Every day we start off with this task, and we move on to two novel symbols which a monkey has never seen before. And he has to learn which means left and which means right by trial and error over a period of trials. So, so during the overtrained condition, I'm plotting the monkey's behavior with respect to a number of trials here. So in the overtrained condition, since he knows the association pretty well, he almost gets it correct all the time. So it's close to 100%, his behavior. And I'm going to show you a neuron, a cerebellar Purkinje neuron. And this is only simple spikes. So this neuron responds to the movement. That's a hand release. So this neuron fires during the hand movement, which is fine. So now once we change the symbols to novel symbol, he starts to guess initially, and then he learns the association by trial and error. So we get a nice sigmoid curve. This is the average learning curve across all sessions. So how does this neuron respond to this change in activity, change in symbol? So this neuron, during the movement, it's fine. It fires the same, pretty much. And after the movement, there's a slight change in activity. And this activity change is shown by this line here. So this is interesting. The first question that would pop to your mind is the movement changing between these two conditions? Because cerebellum is involved in motor error correction. So we track the monkey's hand movement using a high-speed camera. And this is the vertical and horizontal trajectories, and this is the velocity between these two conditions. And we see that the monkey's making the same hand movement over and over again because he's been doing it for like 2 years. So he doesn't want to change the hand movement. So the behavior is similar, the hand movement is similar, but the neuron still changes its activity. So we wanted to know what information does that activity contain. So this yellow trace is actually the average activity in this period, but now we can separate it into correct trials and wrong trials because sometimes he gets it wrong, sometimes he gets it correct. He's guessing in that period. So when we separated that out, we got this. So this is a huge change. And also, this neuron reported the prior wrong trials. So the activity of wrong trials is higher than the overtrained condition, and the activity of correct trials is lower than the overtrained condition. Like, this is pretty significant. Also, the difference, there's one particular epoch when this difference happens, and we call this difference epoch as the delta epoch. So we wanted to know if, are these cells just reporting trial reward outcome? Because we do know that there are granule cells which are one step upstream to these Purkinje neurons do get information about the reward or no reward. So that is pretty unlikely because sometimes, so although he gets it correct most of the time in the overtrained condition, sometimes he occasionally makes errors. And that error is not yet a learning deficit because he's overtrained on the task. So nevertheless, we can separate all the trials in the overtrained condition into correct and wrong trials, and we get this. So here, so in this particular session, while I recorded this neuron, he made about 50 correct trials and 1 wrong trial, so you don't see an error bar for wrong trial. So these neurons are not reporting the reward or no reward, because if the neurons reported the reward, then you would see this line, the correct overtrain, to be with this line, correct learning, and wrong overtrain with wrong learning, but that's not happening. These neurons are reporting the trial outcome during learning, and during overtrain, they don't really care much. So do all the neurons just report wrong trials in this region that I'm recording from? No. So this population of neurons report wrong trials. And we found another population of neurons which reported the correct trials. So these are 2 populations of neurons reporting the prior trials outcome, which is nice to know. And the next question that we had was, does the delta epoch happen just soon after the response, or does it happen anywhere in the trial? This is pretty non-intuitive, but just bear with me for a minute. So here, these are the same neurons that I just showed you. This neuron reporting the prior wrong trial and prior correct trial, but after the movement, after the reward. But we found different types of neurons that had the delta epoch in different epochs of the same trial. For example, this neuron reported the prior wrong trial even before the symbol onset. So this neuron was reporting the previous trial's outcome. And this neuron still reported the previous trial's outcome, but after the symbol onset, and this before the movement. So all these neurons passively reported the prior trial's outcome, and they did not predict the next trial's outcome. Similarly, we find a group of neurons which report the prior correct trial, like similar to these neurons. So these are 8 representative neurons. What happens to the population of neurons? So now I can plot the difference in activity, wrong minus correct, and I can I can sort the neurons based on the time of wrong minus correct, and I end up with this kind of a plot. So here we see each line is a neuron, and each line shows the difference in activity between the prior wrong and the prior correct. And these are for wrong preferring neurons, that's the top column, the top row, and these are for the correct preferring neurons. So we see that the time of difference between the prior wrong and prior correct tiles the entire trial period. So different Purkinje neurons encode this difference in activity in different small time periods, and that difference tiles the entire trial period. So this is great. We are showing that we have neurons that report the prior wrong trial, prior correct trial. Great. So, but if these neurons were to be involved in learning, then we would expect that this activity to change with learning, and that could be a neural correlate of learning. So next question was, what happens through learning? I'm going to show you 2 example neurons, one correct preferring neuron, one wrong preferring neuron in the overtrained condition. In the first third of learning, we already seen that these neurons change their activity. Correct preferring neurons go up, wrong preferring neurons go down. In the second third, the activity still continues to change in the same direction, and that continues to the last third too. So these neurons change their activity in the delta epoch in a linear way with learning. And so these neurons reset their activity and advanced their activity, reset and advance. But we found other groups of neurons, correct preferring and wrong preferring, which reset their activity and then came back to the overtrained. So reset and retract. Similarly, reset and retract. Nevertheless, all these neurons showed a linear change in activity with learning, and that is best explained by this kind of analysis, where I'm plotting the difference in activity in the delta epoch for consecutive trials through the learning. And this is a representative learning curve, and this is one neuron, showing that the activity actually tracks the learning curve with a really good correlation. For the other group of neurons, it happens in the other direction. So to summarize, we have two groups of neurons, two groups of neurons which report the prior wrong trial or the prior correct trial. But this difference in activity actually happens throughout the trial, and it trials the entire trial period. And the activity in that epoch changes with learning linearly. So one simple way to think about such kind of a coding is we already know that about 50 Purkinje neurons map onto a single deep cerebellar nuclear neuron, which is the output neuron from the cerebellum. So if these Purkinje neurons were to encode this activity difference in small epochs, this neuron that is integrating all these activity has a continuous representation of the prior trial's outcome. And that changes during learning. And it is nice to think about it, because this puts cerebellum in a good position in this cognitive loop. And we argue that cerebellum might actually be involved in higher order processes and not just motor adaptation. And I would like to thank these groups for funding, and my mentor, Mickey, and Anna, and the travel grant. Thank you.
We have time for one or two questions. I have one question.
Okay.
So you were only looking at simple spikes in this case,
Yes.
right? And we typically kind of associate the main learning signal coming out of the cerebellum as being the complex spikes. Did you take a look at the complex spikes, and are those being modulated with learning?
We did take a look at it. It's very complicated at this point, so I'm not going to talk about it.
That's what I was hoping, but is it complicated in an interesting way?
Not sure about the interesting part, but we'll see.
Hi, wonderful talk.
Naveen.
I just wanted to ask whether you could possibly speculate on why or the function of that particular representation. Thanks.
These neurons reported the prior trial's outcome, and they are using the trial outcome to learn a new visuomotor association. So at the moment, we're not sure why there are two types of neurons, why some neurons prefer wrong trials and some neurons prefer correct trials. Is that your question?
No, sorry, it wasn't clear. Why it was distributed in that way across the whole trial?
Oh, yeah, we're not sure about that. One hypothesis is that the complex spikes are firing at different time points throughout the trial, and that is why we see a difference in activity in the simple spikes. That could be one hypothesis, but we are still testing that hypothesis.